There is a molecule circulating in the bloodstream of most adults reading this sentence. Not a hormone, not a nutrient, not a pharmaceutical compound introduced from outside. A structural fragment of bacteria that have lived in the human intestinal tract since before recorded history, in numbers that exceed the count of human cells in the body by an order of magnitude. Under normal physiological conditions, this molecule belongs exactly where those bacteria live, inside the intestinal lumen, separated from systemic circulation by one of the most precisely engineered barriers in vertebrate biology. That barrier, the intestinal epithelium, reinforced by tight junction complexes, protected by mucus layers, monitored by an extensive resident immune apparatus, has kept this molecule out of the bloodstream for most of human evolutionary history. The evidence accumulated over the past 20 years suggests that this arrangement is more fragile than was once believed, and that when the barrier becomes even marginally compromised, not through acute infection, not through catastrophic injury, but through the chronic, cumulative pressure of ordinary modern life, this molecule crosses into systemic circulation in small but persistent quantities, continuously. For years, the molecule is lipopolysaccharide. What lipopolysaccharide does at high concentrations is not in scientific dispute. At the concentrations reached during gram-negative bacterial sepsis, a severe, life-threatening systemic infection, it activates a cascade of immune responses so powerful that they can kill the host. Septic shock is, in a physiological sense, the immune system responding with full force to a molecule it evolved to treat as an existential threat. That response is appropriate in context. It is catastrophic at the wrong scale. The hypothesis under examination in this lecture proposes something different in kind, not just in degree. It proposes that chronic low-level circulation of lipopolysaccharide at concentrations orders of magnitude below sepsis thresholds, too low to produce fever or clinically detectable inflammation, quietly and persistently stimulates the same immune recognition system in the same tissues across the years and decades of a person's life, and that this persistent, low-grade stimulation may be one contributing thread in the tangled biology of insulin resistance, metabolic inflammation, visceral adiposity, and cardiovascular risk, the cluster of conditions that now affects, in various forms, the majority of adults in industrialized nations. Whether that thread is causal, whether lipopolysaccharide translocation from the gut drives metabolic dysfunction, or whether it is a marker of metabolic dysfunction that has already begun through other mechanisms, is the central question this lecture will examine. It is a question with genuine clinical and therapeutic consequences. And it is a question that the current state of the evidence cannot fully resolve. That matters, because in a field where the distance between biologically plausible and clinically established is frequently obscured by enthusiasm for a compelling hypothesis, being precise about what we know and what we are still working to know is not merely academic. It is the foundation on which useful clinical and public health reasoning has to be built. Lipopolysaccharide is a glycolipid, a molecule in which lipid and polysaccharide structures are covalently linked, embedded in the outer membrane of all gram-negative bacteria. It is not a toxin in the conventional pharmacological sense, synthesized as a weapon. It is a structural component, as fundamental to the architecture of gram-negative bacteria as cell wall peptidoglycan is to gram-positive organisms. When gram-negative bacteria die and their membranes fragment, lipopolysaccharide is released into the surrounding environment. The molecule is organized into 3 structural domains with distinct biological roles. The lipid A portion forms the hydrophobic anchor embedded in the outer membrane leaflet. It is lipid A that carries the primary immunostimulatory activity of lipopolysaccharide, the structural feature that the innate immune system has evolved to detect with extraordinary sensitivity. Attached to lipid A is the core oligosaccharide region, a branch sugar structure that varies between bacterial species but maintains certain conserved features. Extending outward from the core into the aqueous environment is the O antigen, a highly variable polysaccharide chain responsible for much of the antigenic diversity among gram-negative bacteria and a target of adaptive immune responses. The structural variability of lipid A across different bacterial lineages has important implications that the metabolic endotoxemia literature has not always adequately addressed. Not all lipopolysaccharide is immunologically equivalent. The lipid A structures produced by bacteroidetes, one of the 2 dominant phyla of the human gut microbiome, are significantly weaker agonists of toll-like receptor 4 than those produced by Enterobacteriaceae and other Proteobacteria. A gut microbiome dominated by bacteroidetes may produce substantially different systemic immune stimulation from a given quantity of lipopolysaccharide translocation than a microbiome shifted toward proteobacteria dominant. Dysbiosis. Studies that quantify total circulating lipopolysaccharide bioactivity without characterizing the bacterial origin of that lipopolysaccharide are measuring a composite of molecules with substantially different inflammatory potentials. This is a meaningful source of biological heterogeneity that aggregate observations cannot resolve. The immune system's recognition of lipopolysaccharide is not an evolutionary accident. It represents one of the most deeply conserved features of innate immune defense across the animal kingdom. The toll-like receptor family pattern recognition receptors that detect conserved molecular signatures of microbial life evolved before the divergence of vertebrates and invertebrates. Drosophila melanogaster, the common fruit fly, has functional toll receptors involved in innate immune defense. The human toll-like receptor 4 is the product of over 500 million years of evolutionary refinement of this same recognition machinery. The conservation reflects biological reality. Lipid A is structurally constrained. The same features that make it an indispensable component of gram-negative bacterial membrane architecture are the features that the immune system has learned to detect. Because disrupting lipid A structure would compromise membrane integrity and bacterial viability, gram-negative bacteria cannot easily evolve away from being recognized. The recognition system is, in evolutionary terms, remarkably robust. This evolutionary depth creates the tension at the center of the metabolic endotoxemia hypothesis. The toll-like receptor 4 system evolved in a biological context in which lipopolysaccharide in the bloodstream meant acute gram-negative bacterial invasion. The response it evolved to generate rapid, intense, cytokine-mediated, designed to neutralize bacterial infection before it becomes systemic, is calibrated for episodic encounters with a genuine pathogenic threat. The hypothesis asks, what happens when a signaling system calibrated for acute episodic threat is instead activated chronically, at low amplitude, by molecules trickling from a compromised intestinal barrier rather than from invading bacteria? That is not a question the evolutionary history of the system was designed to resolve. It is a question the contemporary research agenda must. The intestinal epithelium is a continuous single-cell layer composed predominantly of columnar epithelial cells called enterocytes, interspersed with goblet cells, enteroendocrine cells, and Paneth cells that lines the entire interior surface of the small intestine and colon. This layer, in aggregate, has a surface area estimated at approximately 32 square meters, roughly the footprint of a small studio apartment, folded into a structure approximately 7 meters long. It is the interface through which virtually every nutrient the body requires must be absorbed. It is also the primary physical barrier separating the luminal contents of the gut from systemic circulation. The structural competence of this barrier depends critically on tight junction complexes located at the apical margins of adjacent enterocytes. Tight junctions are multi-protein assemblies incorporating transmembrane proteins, including occludin, a family of proteins called claudins, with at least 27 distinct subtypes in humans, and junctional adhesion molecules linked to the intracellular actin cytoskeleton through scaffold proteins, including the zonula occludens family. This architecture allows tight junctions to function not as static physical seals, but as dynamically regulated gates, capable of modulating paracellular permeability in response to physiological and pathological signals. The concept of selective permeability is central to understanding why measuring barrier function is more complex than it might appear. The epithelium is not designed to be impermeable, it is designed to be selectively permeable. Water, electrolytes, amino acids, fatty acids, monosaccharides, and certain immune-modulating molecules cross the epithelium through regulated transcellular and paracellular routes as part of normal physiology. The distinction between normal, regulated permeability and pathological barrier compromise is quantitative and functional rather than binary. Measuring intestinal permeability requires sensitive methods, and changes within the range relevant to metabolic endotoxemia may not produce the classic clinical signs of barrier disruption observed in inflammatory bowel disease or critical illness. Lipopolysaccharide enters systemic circulation from the intestinal tract through at least two distinct mechanisms. In states of significant barrier disruption, inflammatory bowel disease, alcohol-related liver disease, critical illness, severe intestinal infection, paracellular translocation through widened, tight junction gaps is likely the dominant pathway. The clinical elevations of circulating lipopolysaccharide in these conditions are substantial and clinically detectable. At lower levels of barrier compromise, a second pathway appears quantitatively important: the co-absorption of lipopolysaccharide with dietary fat through chylomicron formation. During the absorption of dietary fat, enterocytes assemble large lipoprotein particles called chylomicrons that transport dietary triglycerides and fat-soluble vitamins from the intestinal epithelium through the lymphatic system to the thoracic duct and into systemic circulation. Lipopolysaccharide, being a lipid-anchored molecule, can be incorporated into assembling chylomicrons during this process, bypassing the tight junction barrier entirely by using the same transcellular pathway through which dietary fat is absorbed. Multiple studies have demonstrated postprandial increases in plasma lipopolysaccharide that follow a time course consistent with chylomicron kinetics rather than with acute paracellular leak. And the magnitude of these increases correlates with the fat content of the meal consumed. Which environmental exposures compromise the tight junction barrier sufficiently to increase paracellular lipopolysaccharide translocation? The evidence implicates a specific and familiar set of exposures. Dietary patterns characterized by high intake of ultra-processed foods, defined by high concentrations of refined carbohydrates, food-grade emulsifiers, saturated fat from industrially processed sources, and synthetic additives combined with low dietary fiber intake are associated in both animal models and human observational data with reduced expression of claudin and occludin proteins in the intestinal epithelium, reduced microbiome diversity, and increased markers of intestinal permeability. Several emulsifiers used widely in processed food manufacturing, including carboxymethylcellulose and polysorbate, 80, have been shown in animal models to directly alter the mucus layer, protecting the epithelium from luminal bacterial contact at concentrations comparable to those consumed in processed food diets. Sleep insufficiency, even partial, defined as 5 to 6 hours per night, sustained across weeks, has been associated in controlled human studies with increased circulating markers of intestinal permeability and elevated lipopolysaccharide binding protein. The mechanism likely involves glucocorticoid and sympathetic nervous system activation under sleep-deprived conditions, both of which alter tight junction regulation and gut motility in ways that reduce barrier competence. Chronic psychological stress activates the hypothalamic pituitary adrenal axis and the sympathetic adrenal medullary system, producing sustained elevations in cortisol and catecholamines. Both classes of mediator alter intestinal tight junction physiology, gut motility patterns, and the composition of the intestinal microbiome. Human studies examining gut permeability in individuals with high perceived stress or major depressive disorder have found elevated markers of intestinal permeability compared to low-stress controls. The stress-gut barrier connection is bidirectional. A compromised barrier alters the gut-brain signaling axis through mechanisms that may amplify stress-related physiological responses. The convergence of these environmental disruptors is not incidental. Ultra-processed dietary patterns, sleep insufficiency, and chronic psychological stress are not three independent exposures that happen to affect the gut barrier. They are the defining environmental profile of metabolic risk in industrialized populations. They drive insulin resistance, visceral adiposity, dyslipidemia, and cardiovascular risk through multiple well-characterized pathways that do not require lipopolysaccharide as an intermediary. CD14 concentrates lipopolysaccharide at the cell surface and presents it to the signaling complex, comprising toll-like receptor 4 and its co-receptor MD2. MD2 makes direct physical contact with lipid A and undergoes conformational change upon binding, driving toll-like receptor 4 dimerization and initiation of intracellular signaling. The intracellular signaling initiated by toll-like receptor 4 dimerization proceeds through two principal pathways distinguished by their adapter proteins. The first pathway, mediated through the adapter protein called myeloid differentiation, primary response protein 88, initiates rapidly and culminates in activation of the nuclear factor kappa B transcription factor. The mechanism involves sequential recruitment of adapter proteins, activation of interleukin-1 receptor-associated kinase family members, and ultimately activation of a multi-protein complex called inhibitor of nuclear factor kappa B kinase, which phosphorylates and targets for degradation the inhibitor protein that normally sequesters nuclear factor kappa B in the cytoplasm. Released from its inhibitor, nuclear factor kappa B translocates to the nucleus and drives transcription of a large gene set, including tumor necrosis factor alpha, interleukin-1 beta, interleukin-6, interleukin-8, and cyclooxygenase 2. A second signaling pathway, mediated through different adapter proteins and involving activation of interferon regulatory factor 3, produces type 1 interferons and contributes to antiviral as well as antibacterial responses. This pathway is slower than the nuclear factor kappa B arm and is responsible for a distinct set of gene expression changes that overlap with, but are not identical to the first pathway's outputs. Tumor necrosis factor alpha is the canonical effector cytokine of acute inflammatory defense and is central to the metabolic endotoxemia hypothesis. It is a trimeric protein secreted by activated macrophages that signals through two receptors expressed on virtually every nucleated cell in the body. Interleukin-6 occupies a uniquely complex position in metabolic biology. Acute, transient elevations of interleukin-6 as occur during exercise. When contracting skeletal muscle releases it, as a myokine are associated with metabolically beneficial effects, including enhanced fat oxidation, improved insulin sensitivity in the post-exercise window, and stimulation of glucagon-like peptide 1 secretion. Chronic, sustained elevations of interleukin-6 as observed in visceral adiposity, metabolic syndrome, and inflammatory states are associated with a fundamentally different biological profile, hepatic insulin resistance through activation of suppressor of cytokine signaling 3. Its occurrence, extent, and metabolic consequences in the context of chronic metabolic endotoxemia in living humans are incompletely characterized. Whether tolerance attenuates the metabolic effects of chronic, low-grade lipopolysaccharide exposure and whether attenuation, if it occurs, is protective or itself pathological, is a genuinely open question that the current evidence cannot answer. The framework for studying lipopolysaccharide in human metabolic disease was substantially shaped by a 2007 paper by Patrice Canny and colleagues, published in the journal Diabetes. Using mouse models, they demonstrated that continuous subcutaneous infusion of lipopolysaccharide at concentrations designed to mimic the range observed in human metabolic syndrome, what they termed metabolic endotoxemia, produce the full phenotype of diet-induced metabolic syndrome, weight gain, adipose tissue inflammation, insulin resistance, and elevated circulating inflammatory markers. In the absence of infection and without the dramatic clinical presentation of acute endotoxemia. The paper was conceptually influential because it provided a mechanistic demonstration that lipopolysaccharide at subclinical concentrations could, in a controlled experimental setting, produce metabolic disease features. It also introduced a research vocabulary, metabolic endotoxemia. Endotoxemia-induced inflammation that organized the field. Translating this framework to humans requires navigating several layers of methodological difficulty that are not always transparently acknowledged in the literature. The measurement problem is foundational. Lipopolysaccharide in human plasma is technically challenging to quantify with precision. The Limulus amoebocyte lysate assay derived from the blood cells of horseshoe crabs, originally developed for pharmaceutical sterility testing, detects lipopolysaccharide bioactivity rather than molecular concentration, and its sensitivity to variations in sample preparation, plasma matrix effects, and lipopolysaccharide structural variants makes interlaboratory comparison unreliable. Lipopolysaccharide in plasma binds extensively to lipoproteins, particularly high-density lipoprotein, which appears to have a physiological lipopolysaccharide neutralizing function, and this binding masks assay signal while simultaneously reducing biological activity. The result is a primary exposure variable that is both difficult to measure and imperfectly correlated with the biologically active fraction that actually engages toll-like receptor 4. For these reasons, many human studies use lipopolysaccharide binding protein as a surrogate exposure marker. Lipopolysaccharide binding protein is a stable, hepatically produced protein whose plasma concentration is more reliably measured than lipopolysaccharide itself. It is induced by lipopolysaccharide exposure and reflects integrated exposure over days to weeks rather than instantaneous circulating concentration. However, lipopolysaccharide binding protein is also itself an acute phase reactant. Its synthesis is upregulated by the same inflammatory cytokines, interleukin-6, tumor necrosis factor alpha, whose production it facilitates. This creates the possibility of a confounding loop. Elevated inflammatory markers in metabolic disease may elevate lipopolysaccharide binding protein independently of increased lipopolysaccharide translocation, making lipopolysaccharide binding protein an imprecise surrogate for gut-derived lipopolysaccharide exposure specifically. With these measurement caveats explicitly acknowledged, the observational associations in human data are consistent and have been replicated across populations. Elevated circulating lipopolysaccharide or lipopolysaccharide binding protein has been associated with obesity. Insulin resistance, assessed by homeostatic model assessment. Type 2 diabetes. Non-alcoholic fatty liver disease, assessed by ultrasound and liver enzyme elevation. And markers of cardiovascular risk, including elevated high-sensitivity C-reactive protein and carotid intima media thickness. In cross-sectional studies, some prospective cohort data suggests that elevated baseline lipopolysaccharide binding protein predicts incident metabolic syndrome and type 2 diabetes after adjustment for established risk factors, meaning that elevated circulating lipopolysaccharide in non-alcoholic fatty liver disease may reflect reduced clearance rather than increased translocation. In each case, the metabolic condition plausibly elevates circulating lipopolysaccharide as a consequence, not a cause. Cross-sectional data is structurally unable to resolve this directionality. Experimental human endotoxin infusion models provide a different evidentiary angle. Multiple groups have administered low-dose intravenous lipopolysaccharide at concentrations designed to produce plasma levels within the metabolic endotoxemia range to healthy human volunteers under controlled conditions and measured downstream metabolic effects. The mechanistic connection between inflammatory cytokine signaling and insulin resistance is one of the better characterized relationships in metabolic biology and deserves careful attention as an independent line of evidence regardless of the specific question of lipopolysaccharide causality. The central molecular lesion in insulin resistance at the cellular level is impaired phosphorylation of insulin receptor substrate 1 on tyrosine residues. Under normal insulin signaling, the insulin receptor, a transmembrane tyrosine kinase, autophosphorylates upon insulin binding and subsequently phosphorylates insulin receptor substrate 1 on specific tyrosine residues. This creates docking sites for the regulatory subunit of phosphatidylinositol 3 kinase, initiating a signaling cascade that ultimately drives translocation of glucose transporter 4 to the plasma membrane in insulin-sensitive cells. Glucose transporter 4 translocation is the mechanism by which insulin stimulates glucose uptake into skeletal muscle and adipose tissue, the two primary sites of postprandial glucose disposal. Inflammatory cytokines interrupt this cascade at the level of insulin receptor substrate 1 through a competing phosphorylation event. Tumor necrosis factor, alpha, and interleukin-1 beta activate serine-threonine kinase cascades, including inhibitor of nuclear factor, kappa B, kinase beta, and c-Jun N-terminal kinase that phosphorylate insulin receptor substrate 1 on serine residues. Serine phosphorylated insulin receptor substrate 1 has reduced capacity to interact productively with the insulin receptor and reduced ability to activate phosphatidylinositol 3 kinase. The result is a blunted downstream insulin signal that fails to produce normal glucose transporter 4 translocation, reducing cellular glucose uptake in response to insulin. This is not a secondary or indirect effect. It is a direct molecular competition between inflammatory and metabolic signaling pathways at a shared regulatory node. Ceramide synthesis provides an additional pathway connecting inflammatory signaling to insulin resistance. Tumor necrosis factor alpha, saturated fatty acids, and other pro-inflammatory stimuli upregulate the de novo ceramide biosynthesis pathway in multiple cell types, including skeletal muscle, liver, and adipose tissue. Ceramide activates protein phosphatase 2A, which dephosphorylates and inactivates protein kinase B, a key kinase in the downstream insulin signaling cascade between phosphatidylinositol 3 kinase and glucose transporter 4 translocation. Elevated ceramide concentrations in skeletal muscle and adipose tissue have been documented in insulin-resistant states and in metabolic syndrome. And experimental reduction of ceramide biosynthesis in animal models improves insulin sensitivity. Whether gut-derived lipopolysaccharide contributes to ceramide accumulation in human metabolic disease through toll-like receptor 4-mediated signaling is biologically plausible but not established by direct human evidence. The visceral adipose tissue compartment merits particular attention in this framework. Visceral fat, the intra-abdominal depot drained by the portal circulation, is not metabolically passive. It is infiltrated by macrophages that, in the setting of adipocyte hypertrophy and cell death associated with positive energy balance, adopt an inflammatory phenotype and produce elevated concentrations of tumor necrosis factor alpha, interleukin 6, and monocyte chemoattractant protein 1. The pro-inflammatory adipokine profile of hypertrophied visceral adipose tissue contributes to hepatic insulin resistance because portal blood flow delivers these cytokines directly to the liver and to systemic inflammatory markers through spillover into the systemic circulation. Visceral adipose tissue also produces reduced concentrations of adiponectin, an anti-inflammatory adipokine that normally enhances insulin sensitivity and hepatic fat oxidation through activation of adenosine monophosphate activated protein kinase. The combination of elevated pro-inflammatory adipokines and reduced adiponectin from visceral fat creates a systemic inflammatory environment in metabolic syndrome that is substantially independent of gut-derived lipopolysaccharide. This independence matters for interpreting the observational associations between lipopolysaccharide and metabolic disease, visceral adiposity, which is itself driven by energy imbalance, produces inflammatory signaling that could sustain metabolic insulin resistance, even in the complete absence of gut barrier compromise. The metabolic syndrome defined clinically by the co-occurrence of visceral adiposity, elevated fasting glucose or impaired glucose tolerance, dyslipidemia, characterized by elevated triglycerides and reduced high-density lipoprotein cholesterol, and hypertension may be best understood as a state in which multiple pro-inflammatory signals from multiple sources converge on the same insulin signaling machinery. Gut-derived lipopolysaccharide, if it contributes to this state, does so within a system already characterized by adipose tissue inflammation, altered hepatic lipid metabolism, skeletal muscle lipid accumulation, and dysregulated adipokine signaling. Isolating the specific contribution of lipopolysaccharide within this convergent system is the core methodological challenge the field has not yet adequately addressed. The intervention evidence bearing most directly on the metabolic endotoxemia hypothesis comes primarily from dietary studies examining microbiome composition, gut barrier function, and circulating inflammatory markers as outcomes. These studies do not typically isolate the lipopolysaccharide pathway specifically. They examine dietary patterns and their downstream biological effects, of which lipopolysaccharide translocation is one among many. Dietary fiber, specifically the diverse fermentable polysaccharides found in vegetables, legumes, whole grains, and fruits, has well-documented effects on gut microbiome composition. Fermentable fibers that reach the colon provide substrate for bacterial fermentation by Bifidobacterium, Lactobacillus, Faecalibacterium prausnitzi, Rosburgia intestinalis, and other species associated with gut barrier competence. Fermentation yields short-chain fatty acids, primarily acetate, propionate, and butyrate that serve as the primary energy substrate for colonocytes, support tight junction protein expression, reduce intestinal permeability in cell culture and animal models, and modulate the intestinal immune environment toward reduced pro-inflammatory activation. Butyrate in particular activates the transcription factor peroxisome proliferator activated receptor gamma in colonocytes and suppresses nuclear factor kappa B signaling. The 2021 Stanford randomized trial, published in Cell, compared high fermented food dietary patterns including traditional fermented dairy products, fermented vegetables, and other lacto-fermented preparations with high fiber dietary patterns in 36 healthy adults over 10 weeks. The primary finding was a consistent increase in microbiome diversity in the fermented food group, accompanied by decreases in 19 circulating inflammatory proteins, including tumor necrosis factor alpha, interleukin 12, and interleukin 17 across all participants in the fermented food arm. The high-fiber group showed no consistent increase in microbiome diversity and no consistent decrease in inflammatory markers over the same period, suggesting that adequate baseline microbiome diversity may be a prerequisite for fiber diversity to produce its expected compositional and functional effects. Whether the inflammatory decreases in the fermented food group reflected reduced lipopolysaccharide translocation, direct immunomodulatory effects of fermented food components, microbiome-mediated changes in short-chain fatty acid production, or other mechanisms was not determined by the study design. Fermented dairy products, traditional kefir preparations in particular, contain live populations of lactobacillus and bifidobacterium species, including Lactobacillus kefiranofaciens, whose genomic continuity with human populations has been documented across at least 3,500 years by archaeogenomic studies of ancient dairy samples. These organisms can, in controlled conditions, modulate tight junction protein expression and reduce markers of intestinal permeability, support mucus layer integrity, and competitively exclude pathogenic proteobacteria that may be a source of more immunostimulatory lipopolysaccharide structural variants. Whether these effects translate into reduced circulating lipopolysaccharide in humans under real dietary conditions, as opposed to controlled supplementation models, is an open question with biological plausibility and insufficient direct evidence. The critical constraint in interpreting dietary intervention research for the purposes of this hypothesis is the impossibility of attributing observed metabolic improvements to lipopolysaccharide reduction specifically. Any dietary intervention that improves microbiome composition simultaneously alters short-chain fatty acid production, bile acid metabolism, gut motility, enteroendocrine peptide secretion, intestinal immune function, and the composition of the systemic metabolome. Each of these changes has independent metabolic consequences. The attribution problem isolating the contribution of lipopolysaccharide reduction to observed outcomes would require experimental designs that are difficult to execute in human subjects and have not yet been undertaken. Before reaching conclusions, it is worth being explicit about what each level of evidence examined in this lecture does and does not establish, and where the gaps between levels are most consequential. Biological plausibility refers to the degree to which a proposed mechanism is consistent with established biological principles and experimentally characterized molecular interactions. Lipopolysaccharide, as a contributor to metabolic inflammation, has very strong biological plausibility. The toll-like receptor 4 signaling pathway is real and well characterized. Its cytokine outputs tumor necrosis factor alpha, interleukin-1 beta, interleukin-6 are well-established mediators of insulin signaling disruption. The gut barrier can be compromised by documented environmental exposures. Biological plausibility does not establish that a mechanism operates in vivo at relevant concentrations and durations. It establishes that the hypothesis is mechanistically coherent and worth investigating. Mechanistic evidence refers to demonstration, in controlled experimental conditions, that a proposed mechanism can produce the proposed effect. The acute human endotoxin infusion studies establish mechanistic evidence. Intravenous lipopolysaccharide can produce transient insulin resistance in humans at concentrations within the metabolic endotoxemia range. The mouse model studies establish mechanistic evidence. Subcutaneous infusion of lipopolysaccharide at low concentrations can produce metabolic syndrome features. Mechanistic evidence is necessary, but not sufficient for establishing clinical relevance. The experimental conditions in which mechanistic evidence is gathered: intravenous administration in healthy volunteers, continuous infusion in rodents, differ substantially from the conditions of chronic gut-derived translocation in humans living with metabolic disease. Observational association refers to consistent statistical relationships between measured exposure and outcome in human populations. The lipopolysaccharide literature has substantial observational evidence, with cross-sectional and some prospective data linking circulating lipopolysaccharide and lipopolysaccharide binding protein to insulin resistance, visceral adiposity, non-alcoholic fatty liver disease, and cardiovascular risk markers. These associations are consistent and replicated across populations. They support the hypothesis. They do not establish causation. Cross-sectional associations are structurally susceptible to reverse causality, and adjustment for confounders in observational studies of this complexity is necessarily incomplete. The confidence that can be derived from observational association alone is substantial but bounded. Randomized controlled human trial evidence designed to test the specific effect of an intervention that reduces circulating lipopolysaccharide on metabolic outcomes represents the evidentiary level most needed and least available in this literature. The dietary intervention studies reviewed in Section 7 are valuable but do not satisfy this evidentiary standard because they alter multiple biological variables simultaneously and cannot attribute observed effects to lipopolysaccharide reduction specifically. A properly designed randomized trial would require a validated, stable method for quantifying circulating lipopolysaccharide or a validated functional surrogate. A well-characterized intervention that selectively reduces lipopolysaccharide translocation without substantially altering other biological variables. Adequate statistical power to detect metabolic effects of clinically meaningful magnitude, and follow-up duration sufficient to assess metabolic outcomes beyond acute surrogate biomarkers. Such trials do not currently exist at the scale required to resolve the central question. The current evidential position is therefore as follows. The biological plausibility of the metabolic endotoxemia hypothesis is strong. The mechanistic evidence from experimental models is supportive. The observational associations in human populations are consistent and replicated. The randomized trial evidence needed to establish causal efficacy and clinical therapeutic relevance in humans is insufficient. This is not a critique of the research agenda, it is an accurate description of where the field stands, and the trials needed to advance it are methodologically demanding. Identifying this gap clearly is a prerequisite for the research community's ability to close it. The central question introduced at the beginning of this lecture was not rhetorical. Does circulating lipopolysaccharide initiate metabolic dysfunction? Does it arrive in systemic circulation and begin the inflammatory cascade that ends in insulin resistance, visceral adiposity, and cardiovascular risk? Or does it appear in circulation as an amplifier and marker of metabolic dysfunction that has already begun through other mechanisms, through energy imbalance, adipose tissue inflammation, hepatic lipid accumulation with gut barrier compromise, following as a consequence, rather than preceding as a cause? The evidence does not permit a definitive answer to this question as stated. What it permits is a more precise characterization of what is established, what is consistent with available evidence but unconfirmed, and what is genuinely unknown. What is established? Lipopolysaccharide activates toll-like receptor 4. Toll-like receptor 4 activation produces cytokines that interfere directly with insulin signaling at well-characterized molecular nodes. At acutely administered experimental doses, intravenous lipopolysaccharide produces insulin resistance in healthy humans. Gut barrier integrity can be compromised by dietary patterns, sleep insufficiency, and chronic stress that are also well-established drivers of metabolic risk. Circulating lipopolysaccharide markers are consistently associated with metabolic disease in human observational studies. What is consistent with available evidence but unconfirmed? That chronic, low-level lipopolysaccharide translocation from a compromised gut barrier contributes to the development or progression of insulin resistance, metabolic inflammation, and cardiovascular risk in humans over years of exposure. This pathway is mechanistically coherent, observationally consistent, and has biological plausibility. It has not been demonstrated in adequately powered randomized trials with validated biomarkers and sufficient follow-up. What is genuinely unknown? Whether immune tolerance to chronic low-level lipopolysaccharide exposure develops in humans, and if so, whether it is protective or pathological. Whether the lipopolysaccharide structural variants produced by the microbiome composition characteristic of metabolic syndrome are more or less immunostimulatory than those characteristic of healthy microbiome composition. Whether reducing circulating lipopolysaccharide, specifically independent of the dietary and lifestyle changes that would accomplish that reduction, improves metabolic outcomes. One conclusion is more robust than the causal question allows. The environmental exposures that compromise gut barrier integrity, ultra-processed dietary patterns, sleep insufficiency, chronic psychological stress, physical inactivity are among the most thoroughly documented drivers of metabolic risk in the contemporary epidemiological and clinical literature. They are not risk factors for metabolic disease because they increase lipopolysaccharide translocation. They are risk factors for metabolic disease through multiple converging pathways: adipose tissue inflammation, altered hepatic metabolism, skeletal muscle lipid accumulation, endocrine dysregulation, autonomic nervous system effects that do not depend on gut barrier compromise at all. If gut barrier compromise and lipopolysaccharide translocation are additional mechanisms through which these exposures produce metabolic disease, that increases the public health relevance of addressing them. It does not create a new category of intervention target. It deepens the case for what the evidence already supports, addressing the upstream environmental drivers directly. The metabolic endotoxemia hypothesis is compelling. Compelling hypotheses in complex biological systems are not automatically correct. The appropriate response to a compelling hypothesis with incomplete human causal evidence is not uncritical adoption, and not dismissal, but continued rigorous investigation with experimental designs adequate to the question's difficulty. The gut barrier is not dramatic when it fails. The conditions that compromise it are not dramatic either. They are ordinary, the accumulated effects of sleep patterns, dietary choices, stress exposures, and physical activity that characterize modern industrialized life. Their metabolic consequences accumulate silently, over years, through mechanisms that intersect with lipopolysaccharide translocation in ways the research community is still characterizing. That is precisely why the question the metabolic endotoxemia hypothesis is asking is worth the rigor it has not yet fully received.